Good morning, all. Welcome to our official YouTube channel, SRNBGNR Government Arts and Science College, Khammam. Myself, Dr. B. Satish Kumar, Assistant Professor in Biochemistry. In today's video lesson, I am going to explain about some molecular biology topics. This molecular biology paper, which is present in final year uh, syllabus of final year students, that is semester 6, paper 7, and it is core paper. So in this total paper, molecular biology, we will study about the basic molecular biology like replication, transcription, translation, post translational modifications, all those things, and along with them regulation of gene expression and RDNA technology also. So we have 4 different units covering molecular biology as well as RDNA technology, that is biotechnology also. Today's paper, in today's video lesson, we are going to study the replication process. Let us see the topics which are to be covered in unit 1 of this paper 7. This unit 1 comprising of replication and transcription, two process, and the first subtopic in this unit is just fundamentals about nucleic acids, organization of genome in prokaryotes and eukaryotes, experimental evidences to prove nucleic acids as genetic material, nature and structure of gene. These are just basic topics. And second topic is replication, third topic is subtopic is transcription, and fourth subtopic is different types of RNA polymerases of eukaryotes and post transcriptional modifications also we will study in this unit. To start with, the first topic is some fundamentals of nucleic acids. Before going to study about the replication process, we have to know some basics about the structure of nucleic acids. In semester 2, you have learned about the double helical structure of DNA. So what is DNA and what is RNA, and what is the structure of DNA, and what are the different types of RNA molecules, all you have studied in the second semester. The knowledge of double helical structure of DNA is prerequisite to understand the replication process. So that is why I will explain briefly about the structure of DNA. So as we all know that DNA is the genetic material of almost all organisms. There are only few viruses which use RNA as genetic material, except those viruses which are known as retroviruses. Remaining all organisms will have DNA as genetic material. So DNA is a double stranded structure which is also a polymer of nucleotides, deoxynucleotides. The main difference between DNA and RNA is DNA uses deoxyribose sugar in its nucleotide structure, whereas RNA contains only ribose sugar. Here in double helical structure, originally the double helical structure was proposed by Watson and Crick. And in the double helical structure, we will have two different strands. Those two strands run in opposite direction, anti-parallel direction. So one runs from 5 prime to 3 prime direction, and another runs from that also runs in 5 prime to 3 prime direction, but opposite to the first strand. So anti-parallel strands will be there, and each and every the monomer of DNA is a deoxyribonucleotide. So suppose this is a unit, a monomeric unit, which contains 5 prime phosphate, a deoxyribo sugar, and a nitrogenous base. And as we all know, that DNA contains only 4 different types of bases: adenine, guanine, thymine, and cytosine. So the most important thing in DNA structure is that adenine base pairs with thymine and guanine base pairs with cytosine. Adenine base pairs with thymine by means of 2 hydrogen bonds, whereas guanine base pairs with cytosine by means of 3 hydrogen bonds. Adenine and guanine, these two are called purines, which are two membered rings. So two rings will be there in the structure of adenine and guanine. But in case of thymine and cytosine, these are pyrimidines. They have only one ring structure. So the size of purines is more when compared with the pyrimidine. So therefore, a purine always base pairs with a pyrimidine. So that is why the distance all the between the two strands will be equal from top to bottom. So there is no uh, mm, uh, there is uniformity in the double helical structure. So here you can see that size of the A is more, and A and G, the size of A and G is more than C and T. So that is why the distance between these two strands will be always equal, because a purine is interacting with pyrimidine. No purines will interact with each other, no pyrimidines will interact with each other. So this is what the structure of the double helical structure of DNA. So the main important thing is A interacts with B and G interacts with C. So these two are called base pairing rules. So these base pairing rules are very much essential for replication process. So we can, if we know the sequence of one strand, we can easily predict the sequence in the other opposite strand. So because we know the base pairing rules, that is A base pairs T and G base pairs C. And so this is about the double helical structure of DNA. In the first topic of this unit is nucleic acids as genetic material. So there are two different experiments which proved that the nucleic acids are the the original genetic material. Neither proteins nor carbohydrates nor lipids are the genetic material. So only the nucleic acids acts as genetic material. So genetic material is which contains genes that codes proteins. So all the information for coding different proteins is present in the genome or genetic material. So therefore, the entire information of life processes is present in the genome. So that genome contains DNA as genetic material. So nucleic acids as genetic material. So we have four different types of biomolecules that we studied in the first year: carbohydrates, lipids, proteins, and nucleic acids. So among the four, which one acts as the genetic material? So to prove that nucleic acids acts as genetic material, two experiments were conducted. The first experiment is Griffith bacterial transformation experiment. Frederick Griffith, he is the scientist which proved that DNA is the actual genetic material. So in fact, he found out that some material is there that is responsible for the transfer of genetic information. So the genetic information is present within a, a molecule. So in this experiment, first he used pneumococci bacteria, where the pneumococci bacteria is having two different strains, rough strain and smooth strain. So the rough strain is a virulent strain. So that rough strain means when we grow the pneumococci bacteria on agar medium, that gives colonies which appears a little bit rough in nature. And when we grow the smooth strain on the medium, that gives very smooth colonies. The appearance will be very smooth. So that is why they named that bacteria as rough strain and smooth strain. But in fact, the smooth strain is virulent strain that causes pneumonia to the host cell, the infected organism. Whereas the rough strain is avirulent. So if we inject the rough strain to any individual or any animal or animal model, so nothing will happen. But if we inject the smooth strain, that causes disease. So therefore, first of all, he injected the rough strain to mice. The mice survived because that is a virulent bacteria. And he has taken another batch of mice which were injected by smooth strain. So after few days, the mice developed pneumonia and died. So that means the virulent phase, the smooth strain is virulent strain. And in the next batch, next time, he he has taken the smooth strain and heated to some extent so that the bacteria dies. So once the bacteria is dead, he injected the dead bacteria, heat killed bacteria to mice. After few days, the mice survive because the bacteria is not in living condition, that is already dead. So the dead bacteria is unable to cause virulence to the infected animal. So that is why the mice survived. But in the last stage, he combined that rough strain, which is a virulent strain, and heat killed smooth strain, which is also a virulent because that bacteria is already dead. So he mixed both these samples and he injected to the bacteria. Injected to mice. So uh, because because the rough strain is avirulent and heat killed smooth strain, that is also avirulent, there should not be any pneumonia and mice should survive in this case. But to his surprise, he found that the mice died by getting pneumonia. So he explained explained these experimental results that the heat killed smooth strain, the genetic information for the virulence is transferred from heat killed bacteria to rough strain so that the rough strain becomes smooth strain. So he isolated bacteria from the dead mice and he cultured it. So he found out that the dead mice contained smooth strain. So it is only possible when the rough strain becomes a smooth strain. So rough strain became smooth strain and that information came from the dead bacteria. So this is called transformation. Transformation means the rough strain is transformed to smooth strain. So he explained that some information in the form of some molecule is transferred from heat killed bacteria smooth strain to rough strain. So that made or transformed rough strain to smooth strain. So he called it as transforming principle. Later, Avery, MacLeod, and McCarthy, these are the three scientists who injected purified carbohydrates from heat killed strain, purified lipids, purified proteins, and purified nucleic acids to the mice. So, only those mice which injected by purified nucleic acids are died. Purified nucleic acids plus rough strain. So that caused the transformation. So in that way, they found out that nucleic acids are the principal molecules which carries the genetic information. So that genetic information from the heat killed smooth strain to rough strain was transferred. Therefore, the rough strain is transformed into smooth strain and caused the pneumonia, resulting in the death of mice. So this is the first experiment which gave the for evidence that nucleic acids are the genetic material. Another experiment that proved nucleic acids as genetic material is Hershey and Chase experiment. In this experiment, the investigators used radio labeled molecules. In one set of experiment, they used radio labeled bacteriophages. That bacteriophages protein coat is radio labeled here. So this red color shows the protein coat of bacteriophage that infects E. coli cells, and here the bacteriophage which infects the E. coli cell, in its structure it contains a protein coat that encapsulates its genetic material, the bacteriophage genetic material. So in the first set of experiment, they used protein coat, radio labeled protein coat only. So therefore, the nucleic acid inside that protein coat is non-radio labeled. And in another set, instead of protein coat, here the nucleic acids are radio labeled. So in one set, protein coat is radio labeled. In another experiment, the nucleic acids are radio labeled. So here they allowed these two different kinds of bacteriophages to infect their host cells, and after successful infection, during their life cycle, the bacteriophages inject their nucleic acids into host cell. So after injecting the host cell, they took these two different samples separately and blended them. So blending means vigorous shaking. Because of that, these the attached protein coat upon the cell will be released. So the protein coat which is outside the cell still attached to the cell membrane get liberated or dissociated from the cell membrane. So after blending, they took those samples and centrifuged. So during the centrifugation, because the cells are heavy, then the protein coats they will form separate bands, and in the supernatant, the protein coat will be collected in both the cases. So now they checked the radioactivity in both the sample. In the first case, they found after centrifugation they found the radioactivity in the supernatant, not in cells. Okay. And in the second case, what happened, they found the radioactivity inside the cells, not in the supernatant. So therefore they concluded that genetic material, the nucleic acids, are entering into the cells, and the protein coat it is not entering into the cell, and it is still outside. And when we continue the life cycle of the bacteriophage inside the bacteria, what happens, the E. coli from the E. coli cell several virions will be produced. So that means the nucleic acids which entered into the cell are responsible for the production of several virions. So that explains, confirms the nucleic acids or the genetic material, because the protein coat is no longer involving in the cycle of the virus. So this is another experiment which proved the nucleic acids are the genetic material, not the proteins. So with this, we have covered the two experiments which proved nucleic acids as genetic material. Now let us go into the details of replication process. Before starting the replication process, we will discuss about what is replication. So replication means preparation of copies. Here, if we take any cell, a parent cell, in this case a parent cell, that is a parent cell. So after DNA replication, it enters the cell division and it forms two daughter cells. So for every cell during its life cycle, that that will undergo cell division process, where the two daughter cells will be formed from one parent cell. And they, if we look at into the genetic material of parent cell, that contains a single DNA molecule. In case of bacteria, suppose if we take the bacteria as an example, the parent bacteria will have one DNA molecule. So during the cell division process, what happens, the one DNA molecule becomes two DNA molecules, and after during the cell division, each one DNA molecule uh, will be the part of each daughter cell. So one DNA becomes two DNA, then only the cell will be divided into two daughter cells. So for the cell division to occur, the DNA replication process is very, very important. So DNA replication is nothing but preparation of copy, another copy of already existed DNA molecule. So in DNA replication process, we are going to study about the replication process in prokaryotes. In prokaryotes, like bacteria, the model system is E. coli cell. In E. coli cell, how the DNA replication proceeds and what are the different enzymes and proteins involved in the replication process, All these things we will discuss in the replication process. So before that, there is one experiment that explains the concept of DNA replication. This concept is called semi-conservative mode, semi-conservative model of DNA replication. Semi-conservative model of replication means the the two strands of DNA will be separated during the replication process. On each old strand, a new strand will be synthesized by following the base pairing rules. So that means if we have these two strands in parent DNA, what happened? These two strands will be separated like this, and on each old strand, a new strand will be synthesized. A new strand will be synthesized. So after the division, what happens? This DNA enters one cell, and this will be the part of another daughter cell. So in each and every daughter cell, we will have a DNA, double stranded DNA, but this double stranded DNA contains one old strand and one newly synthesized strand. So this copying, the replication of old strand is very easy because it strictly follows the Watson-Crick base pairing rules. If A is there, then T will be there on the new strand. If G is there on the old strand, C will be there on the new strand. So by following these Watson-Crick base pairing rules, the E. coli cell can synthesize a new strand around each old strand. So two old strands are there. Two old strands are taken as templates, and on each template a new strand will be synthesized by sequentially arranging appropriate deoxyribonucleotides and joining them to each other to form, and that forms a new strand. So there is an enzyme essential for the DNA replication, that is DNA polymerase. So DNA polymerase is the principal enzyme which synthesize a new strand on the old strand by following Watson-Crick base pairing rules. So that is the basic concept of DNA replication. So here we have mentioned that the DNA replication process is semi-conservative model. Semi-conservative mode means semi means of, conservative means conserved. So that means each and every daughter cell will have one old and one new. That is what semi-conservative model. So this is explained by one particular experiment known as Meselson and Stahl experiment. So this is Meselson and Stahl experiment. In the Meselson and Stahl experiment, first of all, those scientists used E. coli cells, and they grown the E. coli cells in N15 heavy isotope containing nitrogen containing medium. So what happens? The nitrogen is present in the bases of DNA. Each and every base contains nitrogen. That is why they are known as nitrogenous bases. So these heavy isotope will be incorporated into newly formed cells, DNA of newly formed cells. So the scientists grown the E. coli culture for several generations on the medium that contained heavy isotope, that is N15, and they extracted the DNA and centrifuged by density gradient centrifugation. So what happens after centrifugation? They got a band. That band is called heavy band because that band is very much nearer to the bottom of the tube. So at the bottom, the density of the medium is very high, and so there is a gradual decrease of density from bottom to top. So that is why if the density is more, if the density of DNA is more, so the DNA travels to the bottom, and if the density is less, the DNA will form a band nearer to the top of the medium on the test tube. or the tube, the centrifugation tube. So that is the main concept here. So here they extracted the DNA which is completely labeled with N15. So we got a band that is heavy band, and then they have taken the same bacteria and grown in N14 labeled medium, light isotope labeled medium, and they grown only for one generation, only single generation, that is for around 20 minutes. So what happens in one generation? So in earlier we have the DNA that contained two strands, and each strand labeled with N15, heavy strands, two heavy strands. Because it follows a semi-conservative model of replication, what happens in the daughter cells after one generation, we will get two daughter cells per each and every cell. So each daughter cell contains one heavy and one light chain. This light chain is synthesized by N14 in the medium, and heavy chain that is inherited from the parent. So that is why what happens now, the density of this hybrid DNA is little bit lesser than the heavy DNA. So that is why it forms a hybrid band that is just above the heavy band. So now we have the hybrid band, and then once again they have grown the same bacterial cell for 20 more minutes. So that means they entered one more cycle of replication. So what happens, this heavy chain involve this heavy chain uh, involved in the replication and forms one heavy and one light chain hybrid, and from this light chain we will get two light chains, because the old one is light chain and new one that is synthesized from N14 is also light chain. The same way we will get two hybrid DNAs and two light DNAs. So when they extracted the DNA and performed centrifugation experiment, they had one band corresponding to hybrid DNA and another bond corresponding to light DNA. So these results are showing uh, showing that the DNA replication is a semi-conservative model of DNA replication. So DNA, the replication process is nothing but a semi-conservative model of replication. That is why we are getting such a pattern during this experiment. So using this experiment, they have concluded that DNA replication process is a semi-conservative mode of replication process. That DNA polymerase is separating two DNA strands, old strands, and a new strand is being synthesized on each old strand in the replication process. And in the next class, we will learn about the process of DNA replication, where we will see the three different steps: initiation, elongation, and termination of DNA replication process. And thank you.